Hello there, I'm Pieter Verboven. I'm from the University of Leuven in Belgium, where I'm working in the Biosystems department in the research group on post-harvest biology and technology. And I would like to invite you to a clinic at the MSC 2024 meeting in Ottawa on the topic of X-ray imaging. The context of the clinic is the following. Biological materials such as fruits and vegetables, as you see on display here, they come in a wide variety of shapes and sizes, but also internal structures. With the internal structure of a biological material, we actually mean the spatial organization of its components. For instance, the different tissues in an apple, as you see on display here. Now, what we particularly see in this image is the fascinating structure of the vasculature tissue that extends throughout the whole apple and can take lengths up to 20 meters in total in a single apple only. Now, this is a very important structure to the apple as it is the pathway to supply the water, the nutrients and the sugars into the apple while it is growing on the tree. Now, if we take a small piece of tissue within this apple, let's say located here in the middle of the apple, and we again zoom in onto a smaller spatial dimension, for instance the microscopic scale, then we start to see even a totally different structure. We still see one of the vasculature strands here in green running through this piece of apple tissue, but we also start to see individual cells in yellow that are clumped together, and in between the cells we see intercellular spaces. Now, this is also an important structure, of course, because it determines how the water and the sugars and the nutrients are distributed from the vasculature into these individual cells, but also how gases that are important for the respiratory metabolism of the tissue, how they are exchanged between the intercellular spaces and the cells. So the internal structure of a biological material is manifested at many different spatial scales. Also, we recognize that at each spatial scale, the structures are quite complex and highly three-dimensional. So if you want to understand better these structures, which is very relevant for understanding the physiology of the biological material, we need techniques to visualize these structures. Once we have these structures and we can quantify them, we can use them in, for instance, modeling, as an example is given here, so that we can see how these structures are affecting the transport of water, of gases, which are all very relevant for the physiology of the material. Let me give you a little bit of background on X-rays. X-rays are, of course, electromagnetic waves, and they are situated in between the ultraviolet range and the gamma ray range, coming with wavelengths in between 0.01 to 10 nanometer, which allows X-rays to penetrate quite well into biological materials. Now, as the rays are passing through the material, the photons of the X-rays are being absorbed, and the rate of absorption, let's say, is dependent on the atomic number of the elements in the material. So if you have heavier elements, you will have more absorption. If you have lighter elements, then you will have less absorption. And also, of course, the amount of elements, so the density of the material, plays a role in the rate of absorption. If a material is thin, you will have less absorption. If it is thicker, you will also have in total more absorption. So in this way, you can make contrast on a detector which captures the X-rays that have managed to pass through the material. Then finally, X-ray imaging instruments, they come in a wide range of spatial resolutions. So typical medical instruments, as you see here on the left, they have spatial resolutions between 100 and 1000 micrometer, while desktop instruments like the one here that are typical for research purposes, they have better resolution in the order of 1 to 100 micrometer. And then you have really high-end research facilities like these synchrotron facilities that are displayed here. The one in Grenoble is this one, where you can have very high resolutions below micrometers scale, and also very high intensity X-rays, so that you can have very high contrast imaging as well. So let's see how X-rays are being typically used in imaging methods. This is schematically given here in the figure on the bottom left. So typically you would have an X-ray source that is generating the X-rays. They are emitted onto an object that you want to image, which is situated on a sample stage. And then on the other end you have a detector. In this case it is a two-dimensional array detector that is capturing the amount of X-rays that have managed to pass through the object of interest. So of course this generates then a two-dimensional image on the detector, which we call a radiograph. And if we come back to the apple that we had been talking about earlier, then a typical X-ray image would look like this. Yeah? So this is a two-dimensional projection image of the apple, where you see there's some contrast in the image. So the core of the apple is lighter here, because of course this is empty, it's hollow. So X-rays are not being absorbed here, so you see them as a higher intensity on the final radiograph. Also you see a gradient from the center to the edge of the fruit. So in the core of the fruit, or around the core of the fruit, you have more material where the X-rays have to pass through, so you have more absorption there, which creates a darker contrast here. While on the edge you have much less material where the X-rays have to pass through, so in a natural way this gradient in grayscale also gives you a little bit of an idea about the varying thickness of this object. Now if we rotate the sample on the sample stage, as indicated here, step by step, and with every small angle that we rotate we also capture an image. We will generate a lot of images, typically a few hundred or a few thousand images while the object is rotating. And then we have radiographs from multiple angles. And then we can do what we call tomography, which is calculating back the original three-dimensional image of the actual object from these radiographs. It's a mathematical technique which we call reconstruction. And then we can generate an image that is the one on the right here, where we actually see the full three-dimensional shape of the object with all its specific features, including the core in this case, but as in particular images that I showed before, we can then start to resolve the cellular structure or the vasculature tissue and all other aspects that are of interest. So X-ray images as such are just kind of boring grayscale images, where the intensity of a pixel in the image tells you something about the difference in absorption with respect to the neighboring pixels. So we need to still process these images if we want to understand better how they reflect the actual internal structures. So the image processing of X-ray images is not a trivial job, and you need quite some expertise and tools in order to generate images as the ones I show you here, where we see the development of an apple while it is growing from a flower in the initial stages early in the season to a full-grown mature apple at the end of the season. Yeah. So the image processing to obtain these sets of images in the past had been quite long. It took several weeks in order to come up with these final images that give us some insight about how the external shape of the fruit is changing, but also how the internal parts of the fruit are changing, including the vasculature and the core, etc. Of the apple. One way to advance our capabilities of image processing of X-ray images is making use of advances in deep learning. I show you an example here. On the left hand side is a piece of apple tissue that has been imaged by X-ray CT. In gray you see the clusters of cells, and in between the cells you see the dark areas, which are the intercellular spaces. Now the task for image processing here is to find the individual cells here and to segment them out so that we can characterize their shapes and their sizes. Now that's with conventional image processing, this is very hard to do because it's very difficult to find where the interface of the different cells are positioned here in these clusters. Now, making use of a considerable amount of labeled data that we have been collecting in the past, we were able to now train a deep learning network that was also tested on new samples that now automatically is generating the labeled images of the individual cells of this tissue. So on the right hand side here you see each individual cell labeled with a color that represents also the volume of the cell. So now we are able to characterize the cell sizes, their shapes, and have also a 3D complete model of the tissue. To make further advances in X-ray imaging in order to improve contrast and capabilities of visualizing things that are otherwise difficult to visualize, we can make use of some advances in the technologies. One of them is displayed here on the left, is spectral X-ray imaging. In that case, the imaging system is quite the same as conventional X-ray imaging, only the detector is being replaced by a detector that is capable of capturing the photons in the different energy classes so that you can have information or different images for different wavelengths in the X-ray spectrum that you are using. That way it becomes more easy to distinguish features that are being determined by, for instance, elementary composition. A second progress in X-ray imaging is so-called phase contrast imaging, where we not only are looking at the absorption of the X-rays by the object, but also how the X-rays, for instance, are scattered by the object. This can give us information about the microstructure that we are otherwise unable to resolve, because the more the microstructure is present in the sample, the more scattering typically you would have. In that case, we need to modify the system with so-called gratings in order to capture that information. But these systems are under development and some of them are already on the market. Finally, one last challenge that I want to discuss in the clinic is how we can better share data with other researchers. This might be very useful to improve algorithms, for instance, for image processing. We ourselves have made a start with that, so we have a website database available called xplant.org, where we have for a wide range of plant materials, plant organs, already images available for download. Here's an example of a data set of apple, where we not only give you the final processed images that can be used, for instance, for modeling, but also the original data, so you can start with your own image processing on them. And as I said, we have them for quite a wide range of materials now available, and we hope to expand it also in the future. I'm looking forward to meet you in Ottawa and discuss ideas about how to progress the field of X-ray imaging of biological materials. For that, please join me in the clinic on Thursday, June the 6th at 1 p.m.